Welcome to The Explainer. Today we're diving deep into the microscopic world, going undercover to expose the secret tactics of some very clever invaders. We're talking about the bacteria behind some of the most common sexually transmitted infections, and you'll see just how they outsmart our bodies to cause disease.
So let's kick things off with a pretty staggering number. Every single day, over 1 million new cases of curable STIs are diagnosed around the globe. Think about that. And the vast majority of those, they're caused by just a handful of bacteria. So today, we're going to get up close and personal with 3 of the big ones.
First up, we've got Neisseria gonorrhea, the bacterium that causes gonorrhea. And trust me, this is one clever invader. It's a true mastermind of infection with a whole bunch of tricks up its sleeve.
Now, Neisseria gonorrhea is what we call a gram-negative bacterium. All that really means is that in a lab test, it has a thin cell wall that doesn't hold on to a specific purple dye. You might think that makes it sound a bit weak, and, in a way it is. Outside of a human host, this thing is super fragile. But once it gets inside the body, it becomes a formidable pathogen.
So how exactly does it cause so much trouble? Well, it comes prepared with a whole toolkit of what scientists call virulence factors. First, it uses these things called type 4 pili. You can think of them like tiny grappling hooks to grab onto our cells. Then it uses OPA proteins to basically zip itself tightly onto that cell before it invades. It even has this molecular camouflage, a special sugar molecule called silylated LOS that helps it hide from our immune system. As if that wasn't enough, it carries molecular scissors called IgA protease to chop up our defenses and a gatekeeper protein called porin B that helps it resist our immune cells. It's an entire arsenal. Let's zero in on that IgA protease for a second. Immunoglobulin A, or IgA, is the main antibody that acts like a security guard on our mucosal surfaces. It's our body's first line of defense. This enzyme the bacteria releases, it's like a pair of wire cutters, snipping the IgA antibody in half and basically disabling the alarm system before it can even go off.
Gonorrhea is also an incredible master of disguise, and I mean this literally. It steals a sugar molecule, specifically CMP nana, from our own cells and wears it like a cloak of invisibility. This coating makes it resistant to our immune defenses in the bloodstream and helps it go completely undetected.
And here is where things get genuinely scary. Gonorrhea is quickly becoming a superbug. It's developing resistance to the very antibiotics we rely on to treat it. How? Well, it can mutate the target the antibiotic goes after, like the PBT2 gene, so the drug just can't attach anymore. It can also use these little things called efflux pumps to actively spit the antibiotic right back out. And it can even alter its porin B channels to just block the antibiotic from getting inside in the first place. This is a huge public health threat, and we are seriously running out of options to fight it.
Okay, let's switch gears to our next invader, Chlamydia trachomatis.
If gonorrhea is a clever invader, chlamydia is the hidden parasite. This microbe is all about stealth, living and multiplying exclusively inside our own cells.
This lifestyle makes chlamydia what's known as an obligate intracellular pathogen. Obligate means it has to, and intracellular means inside a cell. So it absolutely cannot replicate on its own. It's even nicknamed an energy parasite because it literally steals ATP, the host cell's energy, to power its own growth. It's the ultimate cellular squatter.
And its life cycle is this fascinating 2-phase story of transformation. It starts out as a small, tough particle called the elementary body, or EB. This is its infectious form, built to survive the journey between cells. Once it gets inside a cell, it transforms into a larger active form called the reticular body, or RB. This is the replicating factory. The RB makes tons of copies of itself inside a little protective bubble. Then, those copies change back into the tough little EBs, and the host cell bursts open, releasing a whole new army to go infect more cells. And this whole incredible process happens completely hidden from our immune system.
And the consequences of an untreated chlamydia infection can be absolutely devastating. Certain types, or serovars, can cause a condition called trachoma, and this is the leading infectious cause of blindness in the world. What happens is the bacteria cause chronic inflammation that scars the inside of the eyelid. That scarring makes the eyelashes turn inward and constantly scrape against the cornea with every blink. Over time, that leads to permanent vision loss. And the real tragedy? It's completely preventable.
Finally, let's talk about Treponema pallidum, the bacterium behind syphilis. For centuries, this disease has earned the nickname the Great Imitator because of its uncanny ability to mimic the symptoms of so many other illnesses.
A lot of syphilis's success comes down to its shape. It's a spirochete.
which means it's long, thin, and shaped like a corkscrew. This unique structure lets it literally drill its way through our tissues, making it incredibly invasive. It's so invasive it can even cross the blood-brain barrier to get into our nervous system, and crucially, it can cross the placental barrier, passing from a mother to her unborn child.
And if it's left untreated, the infection progresses through these very distinct stages over years, even decades. It starts with the primary stage, usually a single, painless sore called a chancre. Then comes the secondary stage, with more widespread symptoms like a rash. After that, it can go into a latent or hidden stage for years with no symptoms at all, before re-emerging in a devastating tertiary stage that can cause severe damage to the heart, brain, and other organs.
Syphilis isn't just an imitator, it's a master of evasion. It uses something called antigenic variation, constantly changing its surface proteins, like TPRK, so our antibodies can never get a good lock on it. It also uses phase variation, which is basically flicking its genes on and off to change its appearance. And maybe one of its cleverest tricks, it hides its lipopolysaccharide, or LPS. That molecule would normally scream invader and trigger a massive immune response, but syphilis keeps it tucked away where our body can't see it.
Now, because syphilis can cross the placenta, a pregnant person can pass the infection to their baby. This is called congenital syphilis, and the consequences can be just heartbreaking. Babies born with it can suffer from a whole range of severe health problems, including developmental issues and distinctive physical signs like Hutchinson's teeth and saddle nose. In the worst cases, it can be fatal.
So we've seen cloaking devices, molecular scissors, and all sorts of stealth tactics. It really makes you wonder, right? With such clever invaders, how on earth do we fight back? Well, science gives us the tools. For both gonorrhea and chlamydia, the gold standard is a test called PCR, which is super sensitive at detecting the bacteria's DNA. For gonorrhea, it's also really important to try and grow the bacteria in a lab on a special medium called Thermartin agar. Why? That lets us test which antibiotics will actually work against it. Now, syphilis is the tricky one because we can't grow it in the lab. So for that, instead of looking for the bug itself, we use a test called an enzyme immunoassay, or EIA, which looks for the antibodies our own bodies make to fight the infection.
The takeaway here is pretty clear. These aren't just simple infections. They are complex, biological battles being fought on a microscopic scale. And these bacteria are not standing still. They are constantly evolving new ways to invade, to hide, and to resist everything we throw at them. Which leaves us with the big question. As these microbes evolve, how must our science evolve too, just to stay one step ahead?